Welcome everybody. This is Dr. Gilbert talking to you from Adderhold in the Center for Innovation Instruction, and I am in the recording studio and preparing to deliver our class meeting for September 23rd. Today is September 22nd. This is the day before one of the Jewish New Year's holidays, Yom Kippur, and I'm out on that day tomorrow, and therefore we're going to have an online recording covering today's class content, or the class content for our Wednesday, September 23rd class meeting. So, online presentation, I've got a few announcements for you. First of all, you should be seeing this and know that we are not meeting in person in the classroom on September 23rd. And please let any students who were not in class with us on Monday know that if possible. Of course, I'll post it. It'll be posted on D2L, but just in case, please let them know. How about a little update about our exams? Our Exam 1 grades are, and a key will be posted online as soon as they're ready. We're still grading them. I expect they'll be all completed by Friday morning, and you'll be able to access your grades on D2L. And also, I will have a key posted. If you are in school, you are on campus on Friday, you can come by my office in the afternoon and I can give you your exam then. I will bring all of the exams to class on Monday, September 28th. Okay. Also, for Monday, September 28th, I want to note that our homework 5 is due. Okay, so please fill it out and bring it to class with you. Now, for today's class, I want you to go to the class website, the D2L website, and print out question sheet 6 and use it along with today's online PowerPoint, okay? What we're going to be doing today is starting out with a little review of genetics, mainly because our topic here for today and in the coming section of population genetics uses some of the concepts and vocabulary, and I think we want to get the gears turning by talking about just some of the basic processes, sexual reproduction and heredity, inheritance. So let's get started, and then we'll be moving into our first discussion of microevolution. So here's some familiar Mendelian genetics vocabulary that I would like you to be fresh with. I want your ears to be familiar with it again. When we talk about an individual being homozygous, what we mean is that both alleles that the individual has for a given gene are identical. Heterozygous, both alleles are not identical. This, of course, is for a diploid organism. Now, if both, we want to talk about the kinds of alleles we can have in Mendelian genetics. We can have either dominant alleles or recessive alleles. A dominant allele, of course, masks the trait of a recessive allele, often symbolized with a capital letter, but occasionally there's other notation that's used as well. A recessive allele encodes a trait that's masked by the trait encoded by the dominant allele, typically symbolized by a lowercase letter. In Mendelian genetics, we often, of course, because we're talking about heredity, need to discuss different generations, and we will refer to the parental generation as the P generation or the F0 generation, and their children, their offspring, we will refer to as the F1, and their grandchildren as the F2, etc., etc. Hopefully by now some of this is refreshing your mind, that's pulling up some of the information that you've had in other courses, so that when we talk here about evolutionary biology and genetics, you'll be like, okay, I'm ready. What we're looking at in this slide is what happens during a cross between true breeding plants, and is a classic example of Mendelian inheritance. So in the upper left of this figure, you'll see the F0 generation. You'll see a true breeding pink plant there on the left of the Punnett square, and on the top you'll see a white flowered pea plant. you'll see that pink encodes the dominant allele, big A there. On the left, the recessive allele recessive alleles come from the white pea plant there, little a, little a. When we do a cross of these, we cross-pollinate and we grow the plants up. And while we cross-pollinate and we let the plants go to seed, and then we plant the seeds that form, and we look at the phenotypes of the developing plants, what we see in Mendelian genetics, that all of the offspring in the F1 have the dominant phenotype, in this case pink color flower, and they all have a heterozygous phenotype, meaning that they have one of each of the two alleles. Now, if we let the individuals from the F1 generation there that are heterozygous self-pollinate, so they cross, we can see, as is illustrated here in the bottom left of the slide, the two heterozygous alleles, or the heterozygous gametes merging to yield offspring that have this characteristic 3-to-1 phenotypic ratio and a characteristic 1-to-2-to-1 genotypic ratio. So if you look in the right here of the slide, you'll see that we have homozygous dominant, 2 heterozygous individuals, 1 individual that is homozygous recessive. So this gives us a 1-to-2-to-1 genotypic ratio. And if we look at the phenotypes, you'll see 3 pink, 1 white, a 3-to-1 phenotypic ratio. And by now you're probably saying, yeah, Dr. Gilbert, I remember this well. But that's good. I want you to. Okay, now this is kind of cool. Years after Mendel discovered the principles that we were just looking at, cytologists looking at the process of meiosis under the microscope saw the physical basis for the distribution of traits that Mendel observed. And what we're looking at in this diagram are 2 true breeding parents. In this case, we've got a purple one and a blue one. Here you can see they're homozygous dominant here, big A, little a over here in blue. We see as we go through meiosis 1 that our unduplicated chromosomes here at the top duplicate. So now we have 2 duplicated chromosomes. And at the end of meiosis 1, they segregated, so we actually have haploid cells with duplicated chromosomes, so only 1 copy of each chromosome, but they're duplicated. And as we go through meiosis 2, you'll see that now we have haploid gametes with only 1 copy of each chromosome. So for the homozygous dominant parent, they're all big A. For the homozygous recessive parent, they're all little a. During fertilization, the diploid state is restored and a heterozygous individual is formed. In this case, this would be an example of the F1 as we just were looking at. All of the offspring have this heterozygous big A, little a genotype. All right, so why do we care? Because evolutionary biology involves a fair bit of genetics. Why? When we talk about evolution, we're talking about changes in the allele frequency of a population over time. Why do the changes take place over time? The interaction of populations of organisms with their environment, that's the ecological part of evolutionary biology. But on the genetic side, we need to think about what affects allele frequencies in population. There's several processes that we're going to consider in the next few weeks. One of them that we have considered obviously in quite a fair bit of depth in our class so far has been natural selection. This is one of the processes that can change the allele frequency in a population over time. Now we're also going to talk about gene flow, genetic drift, Last class we talked a bit about mutation and how mutation is the source of raw material for evolution. Next week, when we meet, we'll be talking about non-random mating. Okay, so just to refresh, what do we mean by population? All of the individuals of the same species occupying the same area. What do we mean by an allele? One of two or more slightly different molecular forms of a gene, and they encode for different versions of the same trait. Okay, so this could be curly hair, straight hair. That's an example, that sort of thing. All right, here's a term that we need to think about for this part of our class. We need to think about what we mean, actually a pair of terms, by an individual's genotype, and then what we mean by gene pool. When we talk about a genotype, we're talking about all of the alleles that make up an individual. If you think about it, we all members of the same species all have the same genes, but we vary in the alleles that we have for those genes. So when we compare genotypes, we're comparing all of my alleles, for example, with all of your alleles. And even though we share the same genes, we have different versions of those genes. So we're comparing our genotypes. Now, if we think of all of the genotypes in a population, then we are talking about the gene pool. It's the collective set of alleles that we share in our population. And this is an illustration that I actually think is pretty good. It's pretty basic, but it's helpful. It comes from our Biology 2107 textbook. And what you'll see here there are three genes, X1, X2, and X3. Actually, I take it back. There are three alleles for a single gene, X1 and X2 and X3. And you'll see that each of these ovals here represents an individual organism in a population. This large circle represents the population. And you'll see that the alleles can be mixed and matched in different ways throughout the population. So here we have an individual who's homozygous for X1, and here we have an individual who's heterozygous for X1 and X3. And we can consider, when we consider a gene pool, we're interested in what is the frequency of the allele in the total population. So we could count up the total number of X1, X2, and X3 alleles here and determine what fraction of those were X1, what fraction were X2, what were X3, independent of what the genotypic makeup in the individuals. Doesn't matter whether we're talking about homozygous or heterozygous individuals. We're just interested in the fraction of alleles in the population. So here's our gene pool with a bunch of alleles distributed. Whoops, that little pause there is me looking for the arrow to advance our slide. So I apologize for that. Here we go. The first characteristic feature of microevolution that we want to talk about. Microevolution is the area of evolution where we are talking about the change in allele frequency in populations over time. The first feature of microevolution that we want to talk about is called gene flow. Okay. The first feature of today, as I mentioned earlier, we already talked about natural selection. So the first feature of today's presentation that we want to talk about that affects allele frequencies in a population, gene flow. What is it? It's the physical movement of alleles among populations. So in this diagram, you can see we have one population over here on the left of the slide. On the right, we have another population. And when individuals, organisms, move from one population to the other, they bring their alleles with them, and that affects the frequency of those alleles in the population. When an individual leaves a population, we refer to it as immigration. When individuals arrive in a population, bringing their alleles with them, we call that immigration. So, the physical movement of alleles among populations influences the allele frequency in a population. When the allele frequency in a population changes, our populations are evolving. The next element of microevolution that I want to talk about is called genetic drift. Genetic drift is an interesting type of change in allele frequencies. What's interesting about it is that it's totally random. It doesn't have any, it's a process that's not responding to features of the environment that we think about, such as natural selection being adaptive in some way. Genetic drift is just random change in allele frequencies in a population over time. Randomness, that is just a weird thing to think about being significant in an actual process, but it turns out to be very important. So how do we know when genetic drift is happening? Events that could be described by it just so happened that something happened characterized genetic drift. So here's an example. It just so happened that the baldest guy had the most offspring. And the consequence of this is there are more bald guy alleles in the population. Now, is baldness contributing in some way to the adaptive survival of the population and leading more offspring? I seriously doubt it. Like, is it the sun is shining on some bald guy's head and reflecting light better than somebody with a good head of hair and keeping that individual cool, and somehow that causes more offspring? Really? No, it's not. So it's just a random feature. Okay, it just so happened that there were more of that particular kind of allele in the population because an individual had more offspring than his or her neighbor. Okay, now we'll talk more about genetic drift in this section of the course. It turns out to be very significant as a force in causing evolution in small populations. So you imagine an island that only had 100 individuals, for example. A random change there can be very significant and can cause rapid change to the genetic makeup of the population. All right, let's look ahead here. Mutation, a change in the DNA sequence of a gene. We spent last class period talking about that. It's significant because it is the source of new alleles in a population. Okay. Mutations are the only genuine source of brand new, de novo, from scratch alleles in a population. Gene flow will bring in new alleles from other populations, but we count on mutation to create brand new alleles that had never existed before. And the example we're looking at here is a point mutation. You'll remember that term from Monday, September 21. Here we're looking at a single nucleotide change in the glutamate gene, glutamic acid, codon encoding glutamic acid. The A there in the second position changes by mutation to a C, giving us valine, and that is very significant when we're talking about hemoglobin and the formation of sickled red blood cells and the disease or the condition sickle cell anemia. All right. All of the features that I've mentioned so far are components of a model of evolutionary biology that was developed by three people, one who's not on this slide, but here in the United States we tend to talk about two of them. Their names are Hardy and Weinberg, and at the beginning of the 20th century they each independently drafted some mathematical expressions describing how allele frequencies and hence evolution could take place in a population, and they both came up with these 5 factors as elements of the model. And there's a third guy who's British named Castle. So in Britain they call this the Hardy-Weinberg Castle equilibrium. These mathematical expressions, which we won't be deriving, but we will be talking about in a fair amount of depth during the next couple of weeks, these expressions and their components help us understand whether a population is evolving and why. Okay, now the model has 5 assumptions. Okay, the assumptions are: if there is no selection, no migration, no mutation, no genetic drift, and no non-random mating, i.e. there is random mating, if all of these things are true, then the allele frequencies in a population won't change from generation to generation. In other words, if the allele frequencies don't change, then the population is not evolving. When that's true, another one of the conclusions of the Hardy-Weinberg analysis is that the genotype frequency of the population will fit the formula p squared plus 2pq plus q squared equals 1 when p and q are dominant and recessive alleles for a particular gene in the population. Okay. All right. We're going to pause here just a moment. All right. Back again. The 5 assumptions here you will see are several of the things that we've already talked about. Selection, we can consider, at least in this part of our class, as meaning natural selection. Migration, mutation, genetic drift, we just talked about. By the way, migration is a synonym for gene flow, okay, the physical movement of individuals with their alleles among populations. The only thing we didn't talk about is the non-random mating, and we'll be talking about that in a nearby class meeting. Okay, so why do we care about Hardy-Weinberg equilibrium? Because when a population is in Hardy-Weinberg equilibrium, then we can know that the population is not evolving. In other words, the allele frequencies won't change from generation to generation. Okay, and when a population is in Hardy-Weinberg equilibrium, we can predict the genotype frequency of the population. All right, so at this point, I want each of you to pull out your question set for today's class. Okay, and we are going to start out looking at question 1. The question reads as follows: a population of students at Georgia State has 50 individuals. 32 are homozygous dominant for an allele Q, 16 are heterozygous, and 2 are homozygous recessive. Calculate the frequency of the Q allele in the population. In other words, calculate the allele frequency. All right. So, the first thing we need to do to solve this kind of problem is to count genotypes. What are genotypes? When I am talking about genotypes and we are discussing genotypes, what we mean by that is how many homozygous dominant, heterozygous, and homozygous recessive individuals we have in a population. So in this case, big Q, big Q, big Q, little q, little q, little q. All right, so in our example, we have 32 homozygous dominant individuals, 16 heterozygous individuals, and 2 homozygous recessive individuals. The next step is to calculate the allele frequencies from the data. Let's start out here with the dominant allele. We know we'll start out here with our homozygous dominant individuals. We know that we have 32 of them, and each of them have 2 alleles. So we have 2 times 32 big Q alleles from our homozygous dominant individuals. From our heterozygous individuals, they each have only 1 big Q allele. So we have 16 big Q alleles that come from our heterozygotes. If we do the math here, we end up with 2 times 32 is 64, plus 16 gives us 80 total big Q alleles. There's 100 alleles in the population. How do I know that? There's 50 individuals. They're diploid because they're students at Georgia State. So we have 100 total individuals, alleles in our population. 80 divided by 100 gives us a frequency of 0.8. Now, we can do the same thing for our little q allele. So we only have 2 homozygous recessive individuals. So we have 2 times 2 little q's here, plus 16 little q's that come from our heterozygotes. That gives us a total of 20 little q alleles. 20 divided by 100 is 0.2. Okay, so we can confirm that this is true by doing the calculations for both alleles. If you have questions about this, please bring them with you to class on Monday, and we can do this. I'm talking about Monday, September 28th, and we will take a look at them. I very well may have another problem similar to this. A surprise problem for you. All right. Now, the next step in the assignment is to switch to page 2. Okay. And what we're going to do here in this assignment is 2 things. We're going to select zygotes. We're going to simulate some mating. Okay. This is going to be the least romantic simulation of mating that you have ever done or seen, but we will do it right here in class. And we'll use page 3 of this handout. So we're going to do that for part A. And then for part B, we're going to do a similar type of set of crosses or matings, but we're going to use the Punnett square that's in part B. Okay, and by the end of our class meeting here, you will see why we are doing this. Okay, why are we doing this kind of these 2 exercises? All right, so let's begin part A. Select 25 zygotes using figure 6.3, which is page 3 of this handout. Then 1, determine the genotype frequency, and 2, the allele frequency in the population that will develop from the 25 zygotes. All right, so let's take a look here at the figure, and what you see here are some cute looking little sperms and eggs, and they have either a dominant allele, big A, or recessive allele, little a. And what you are going to do is, to do this exercise, is close your eyes and randomly place your fingers onto the slide or onto the square like this on the handout, and you are creating zygotes by fertilizing eggs with sperm. Of course in simulation, but by doing this, you will get 25 zygotes that have random genotypic makeups. They're either, some fraction will be homozygous dominant, some will be heterozygous, some will be homozygous recessive. Okay, so you can see here, I want you to note at the bottom of this figure, it says that the gene pool has an allele frequency of 0.6 for allele A and 0.4 for that is for allele big A, and 0.4 for allele little a. So once we've done that, we can go ahead and do our calculations. So what we'll do is work with a set of data that I collected by picking in such a manner, and this is the question that I want you to answer. Were the 25 pairs of alleles that you picked for the in-class assignment in Hardy-Weinberg equilibrium or not? So the first thing we need to do is count the genotypes. And what we see in my example is that I had 7 homozygous dominants, 15 heterozygous individuals, and 3 homozygous recessive individuals. Excuse me. And the next thing we're going to do from that information is calculate the allele frequency of the population. Does this sound familiar? It should, because we just did this, right? So you'll see here that I have 2 times 7 homozygous dominant individuals plus 15, that is, I have 2 times 7 big dominant alleles from my homozygous dominant individuals, 15 dominant alleles from my heterozygous, giving me a sum of 29 alleles, okay, that are big A. My population had 25 individuals, so that meant I had 50 total alleles. So 29 divided by 50 is the fraction of the population that is big A, okay? We can do the same thing for little a, the recessive allele, and by doing that, we end up determining that the fraction of little a in the population is 0.42, okay? So, my note here on the side, by the way, is that we can tell already that our allele frequency is different than that in figure 6.3, because as I just mentioned, in figure 6.3, the allele frequency of big A was 0.6 and little a was 0.4, and now we've varied from that to 0.58 and 0.42 respectively a little bit. And note that this data that we're working with here is the data that I picked from using that figure in the previous slide back here. It may differ from the data that you collect. All right, let's move on. Now, while we're here, let's calculate the genotype frequencies for the population. Okay, so far we've calculated allele frequencies. Now we want to know genotype frequencies. So what was the fraction of homozygous dominant, heterozygous, and homozygous recessive individuals in the population? So, how many individuals did we have total in the population? 25. How many are big A, big A? 7 divided by 25 equals, we could look at it like this, 28 out of 100 equals 0.28. We can do the same thing here for the heterozygous individuals. We have 15 out of 25 that are heterozygous. That's the same thing as 60 out of 100. I'm multiplying the denominator and the numerator by 4, and as a fraction we express that as 0.6. Okay, so why do we want to know this? We want to know, in this case, whether we have a violation of Hardy-Weinberg conclusion number 2. What is the Hardy-Weinberg conclusion? If we are in Hardy-Weinberg equilibrium, then our genotype frequency should be in the proportion p squared plus 2pq plus q squared equals 1. Okay, so what are our predicted genotype frequencies based on, how do our genotype frequencies here compare to Hardy-Weinberg predicted genotype frequencies? Okay. Is our population in Hardy-Weinberg equilibrium? If so, the genotype frequencies should be p squared plus 2pq plus q squared equals 1. So what we're going to do here is take our allele frequencies from step 2 back here. Whoops. There they are, 0.58 and 0.42. This is what we calculated. And we're going to take these allele frequencies and we're going to put them into the Hardy-Weinberg expression. So we have 0.58 squared plus 2 times 0.58 times 0.42 plus 0.42 squared. Okay, we just took our allele frequencies and we plugged them in. Big A here for P, little a here for Q, and away we go. Here's what we got. 0.3364, so 0.34 plus 0.49 plus 0.18 equals 1. All right. Now, these are our expected Hardy-Weinberg frequencies. Let me read what it says here in C. In C it says ask. So we're going to ask, are the calculated genotype frequencies from B the same as the observed genotype frequencies from step 3? If yes, we're in Hardy-Weinberg equilibrium. If no, we're not in Hardy-Weinberg equilibrium. So our expected genotype frequencies are here, 0.33, as we said before, 0.49, 0.18. The observed frequencies, okay, this is what we actually observed. Where did we observe it? We observed it back here, are 0.28, 0.6, and 0.12. So our conclusion here is the genotype frequencies are not the same. They're not equal, and our population is not in Hardy-Weinberg equilibrium. So what have we learned? Our analysis shows that both conclusions of both Hardy-Weinberg conclusions were violated. The allele frequencies that we measured were not in Hardy-Weinberg equilibrium. The genotype frequencies that we measured were not in Hardy-Weinberg equilibrium. This means that our population is evolving. Why is it evolving? We don't know yet for sure, but we know it's got to be because one of the 5 factors, one of the 5 assumptions was violated. No selection, no migration, no genetic drift, etc. Okay? If one of those was violated, then our population will not be in Hardy-Weinberg equilibrium. If none of those are violated, our population will be in Hardy-Weinberg equilibrium. All right, so you can mull that over for a little while, and let's move forward to the next part of our problem. If you're at home, maybe you should be watching this with a classmate. Have some popcorn, sit down and work this out together. Excellent. Become friends, why not? All right, let's look at the next part of our worksheet. Complete the Punnett square below, assuming the allele frequency in figure 6.3 of this handout. Then determine the genotype frequency of the resulting zygotes. All right, so what was the allele frequency in figure 6.3 above? It was 0.6 for big A and 0.4 for little a. Let me just flip back there really quick. Yeah, right there. Down at the bottom, you'll see it. It's right there. Okay. How do we do this? Okay, well, if we have a Punnett square grid, as illustrated in the next slide, where we have a grid of 10 by 10, what we can do for 0.6 and 0.4 is to do 6 big A's and 4 little a's. 6 big A's and 4 little a's. We don't have that in our grid. We only have 5. Oh, that's tricky. What are we going to do? Well, we can do 3 big A's and 2 little a's, and that would be the same proportionally as having 6 big A's and 4 little a's. So if you do that, 3 and 2 and 3 and 2, then you can go through and populate. That means fill in your Punnett square with all the corresponding genotypes. Okay. And what we're going to do here is look at the book's example. Okay. And what we're going to see is that if we start out with a Punnett square that's got 6 big A and 4 little a, that's 0.6 and 0.4, and we do all the crosses here, what we're going to end up with is 36 homozygous dominant individuals, 48 individuals that are heterozygous, you can see 24 here and 24 here, and 16 individuals who are homozygous recessive, little a, little a. Okay. Now, what is this? These are the genotypes that we expect to get when every possible cross is done proportionally here using the allele frequencies found in our population, 0.6 and 0.4. Okay. We do the crosses, we end up with this expression. Okay. Now, let's look. So we've done that, we filled it in. Let's go to page 1 of the handout and finish the remaining questions. So, question 2. According to the Hardy-Weinberg equilibrium analysis, under what conditions will no evolution take place? Okay, well, we're busy committing this to memory. We know we will have no evolution if there is no selection, no migration, no drift. We have no non-random mating and no mutation. When those 5 conditions are true, there is no change in the allo frequency in a population from one generation to the next, i.e. there is no evolution. Question 3. According to the Hardy-Weinberg equilibrium analysis, at equilibrium, what is the predicted genotype frequency of a population? It is p squared plus 2pq plus q squared equals 1. So, one of the things that often students find confusing is the difference between allele frequencies and genotype frequencies. Okay, so these are 2 things that I want you to work on keeping track of what they are. Genotype frequency, how many homozygous dominant, heterozygous, homozygous recessive individuals in the population. Allele frequency, the fraction of the population that is each allele, dominant or recessive. Okay, let's get to the key question here, why we did these exercises. Why are the results from parts A and B on page 2 different? Okay, we started with the same figure 6.3, and we used that data to make some calculations about allele frequencies and genotype frequencies. Okay. In the case of the hold on here a second. Let me pause this for a moment so I can work out how to say it nicely. All right, welcome back. Let's consider the 2 things we did and what's different. In the first part of our exercise, we randomly picked zygotes. We randomly formed zygotes by closing our eyes and blindly generating them with figure 6.3. And that gave us an allele frequency, like in my case, 0.58 and 0.42, that perhaps were slightly different than the allele frequencies of the initial population, 0.6 and 0.4. When we did the Punnett square, we generated genotypes using the exact same allele frequency as found in the figure. Okay? So, let me see. There's one piece of information that I would want to add here that I can't quite do because I'm recording. If I was in class, I could write it on the board, or that kind of thing here. If we look back at this slide, we will see we have 36 big A, 48 big A little a, 16 little a little a. Okay, this is the genotype frequency for the gene pool that we looked at here with this allele frequency of 0.6 and 0.4. What I didn't do that I would like to add here is that if you take the allele frequencies given here, 0.6 and 0.4, and you put them into the p squared plus 2pq plus q squared expression, what happens is that you end up with a genotype frequency of 0.36, 0.48, and 0.16. In other words, you end up generating exactly the same distribution of genotypes as you get from doing the Punnett square. Okay, I'll have to show you this briefly when we meet again on Monday, but you can try this. Take that allele frequency and look at the genotype frequencies that you get. If you compare the genotype frequency that you get using these alleles over here, and you compare that with the one that you generated through the calculations like I did above, then what you see is they are different in many cases. The only way they wouldn't be different is if you picked exactly 0.6 and 0.4 randomly when you were doing your selection, your formation of zygotes. Okay. So the question is, what was different about doing it the Punnett square way from doing it the random picking way? And the difference is blind luck. And in the case of pointing at the sheet, we end up with alleles that are selected by randomness. We have genetic drift involved, and as a result, our population is not going to be likely in Hardy-Weinberg equilibrium. The only way it would be is if we ended up with exactly that ratio of 0.6 and 0.4 that we started with. The chances of that are not very high, but it could happen. So what we see is that changing the allele frequency in the one example results in a different answer than compared to the other example. Okay, so randomness becomes important. All right, down here we have a couple of questions addressed. So this is related to what I was just saying. Question, why was there a change in allele frequency even though there was random mating? Don't we need random mating to be in Hardy-Weinberg equilibrium? Yes, random mating is part of Hardy-Weinberg equilibrium, but in the Punnett square, however, all the combinations occur proportionally. No luck is involved. In the random selection, meaning the one we were picking with our fingers, blind luck is involved. And the blind luck that we introduce by picking zygotes simulates genetic drift, part of the Hardy-Weinberg equilibrium model. If there's drift, then the population is not in Hardy-Weinberg equilibrium. And so why then is the Punnett square type combining zygotes not non-random? Okay, it's a little weird there. It's not non-random because it's proportional, but in theory, every egg and sperm in that population had an equal probability of mating, okay, crossing and forming a offspring, forming a zygote with any other sperm or egg. In the true non-random mating, the mates actively select each other, excluding some possible combinations from ever occurring. So it's different than the proportional type of zygote formation that we're seeing in Hardy-Weinberg equilibrium, even with the Punnett square. Okay, so it's sort of a semantic feature about this. What do we mean by random and non-random? We'll get into that a little more detail later in our class. Okay, but in the short run, we can see that we can focus on the difference here by random picking versus not random picking, and it's an important feature of Hardy-Weinberg. So what do you get out of doing this exercise? Well, you get a lot of experience using the terms allele frequency and genotype frequency, some basic calculations that are involved, and familiarity with the elements that feed into the Hardy-Weinberg equilibrium analysis. With that, we're going to sign off. I'll see you in class on Monday, September 28th. Have a great weekend. Work on your homework. Find a study partner and work with them. And I'll have your exam grades ready for you soon. Bye-bye.